So we're now going to go through more detail about light-dependent reactions. Okay, so the big thing that happens in the light-dependent reactions is we are using photosystems. Okay, so these are clusters of pigment molecules, and they're bound to proteins along with a primary electron acceptor. The big thing here is that that's where the chlorophyll is located. Okay, so these are just kind of like clusters of pigment molecules, which is the chlorophyll, and that is what is absorbing the light. There are two photosystems that are involved: photosystem 2 and photosystem 1, and they both absorb slightly different wavelengths of light. Okay, so photosystem 2 absorbs 680 nanometers, and photosystem 1 absorbs 700 nanometers best. I don't know that you really need to memorize those numbers, but photosystem 2 actually occurs first in the light-dependent reaction because it was, the only reason it's named photosystem 2 is because it was discovered second. And then photosystem 1 was actually discovered first. So it sounds a little out of order, but it's because they're numbered 1 and 2 in the order of when they were discovered, but they were discovered out of order. So anyway, so our overview of what happens. This is showing like our membrane here. I just didn't draw in all the phospholipids and everything. We got our stroma outside here, and then in here is inside the thylakoid in our thylakoid space. Okay, and you can think that there's water kind of everywhere, but we'll talk about water in a little bit. Okay, so we have photosystem 2 and photosystem 1, and they reside on the thylakoid membrane. Okay, and so there's also an electron transport chain here as well that we'll talk about in a second. And there's ions kind of all over the place. Hydrogen ions, remember that's what's accounting for different pH levels. So hydrogen ions just kind of float all around. Okay, and this is representing electrons in our different photosystems. Okay, so light energy strikes photosystem 2 first, and what it does is it excites an electron in that photosystem. And when it does, that electron is transferred to the electron transport chain. And what actually happens is that we've just lost an electron, making this photosystem now negatively charged, which it doesn't want to be because it's inside the membrane. So it doesn't want to have that polar charge involved, which we'll talk about that in a minute. Or I'm sorry, I guess I have that next. Okay, so there's kind of two things happening at the same time. So one thing is because of that negative charge, we don't want it to stay negative. So what happens is this water molecule, and remember there's water all over the place, is split, and when it does that, an electron, anytime we break apart molecules, you can think of like electrons just kind of go flying. So the electron from water is taken to the photosystem 2 to put it back to what it was before. But when it did that, it split this water, and now we have oxygen gas. This would happen like 2 at a time until we have O2, because oxygen gas is diatomic. But anyway, and then that oxygen can just leave straight through the membrane because it's small and nonpolar. So at the same time that's happening, this excited electron is going down the electron transport chain. And when it does, it's providing some energy because it's an excited electron. So what it does is it powers a proton pump. And so that excited electron is transferred, and it pumps hydrogen ions out of the stroma into the thylakoid. So it's pumping hydrogens. So we're making it more acidic inside and less acidic outside. Okay, when it does that, it just used up its energy. So now we have our electron at photosystem 1, and it used its energy to pump that hydrogen in. And so what happens is photosystem 2 gets those electrons there are energized as well, because there's light hitting photosystem 2 and photosystem 1. And this time from photosystem 1, that excited electron goes, and instead of that energy being used to pump hydrogens in, it is actually just transferred to NADP plus to make NADPH. And that is going to go on again in the stroma to be used in the Calvin cycle. The rest of what happens to make ATP is because we just pumped a bunch of hydrogens inside our thylakoid. So we have created a proton gradient where we have a lot of hydrogens that have been pumped inside. And so that creates, basically you can think of it like a dam where we have blocked up the water, we have stored it up. Now we have a whole bunch of hydrogens that we can release through the dam at a controlled rate so that we can get energy. So the cells do that also. So these hydrogens, they can't go straight across the membrane because they're charged. They have to go through something, and there is a protein called ATP synthase, and when these hydrogen ions can pass through them, it basically smashes a phosphate onto ADP, making ATP. And so when it does that, those hydrogens naturally do this because they're naturally wanting to go from high concentration to low concentration. So that is what's happening in the light-dependent reactions. This is another way of showing that instead of having all the pieces moving, it's just one drawing. So we've got light energy coming in. We have photosystem 2, that electron. And there's other enzymes and there's other proteins involved in this that we don't have to get into. This is showing like a couple different enzymes or proteins that are embedded in the membrane that the electron gets transported to, but we don't need to know that. Just that it's transferred along to different proteins, and these hydrogens are pumped into the thylakoid. Okay, and then it goes to photosystem 1, more light, energizes, and then that energized electron turns NADP plus into NADPH. Now that we have a bunch of hydrogens in here, these hydrogens go through ATP synthase, and when they do that, they turn ADP into ATP. And then at the same time, at the very beginning, when we lost that electron, water is turned into oxygen gas and actually more hydrogen ions, which again adds to that hydrogen ion gradient that we've made, that proton gradient inside the thylakoid. So that is what's happening in the light-dependent reactions. Big thing is we are using the light to pump hydrogens in, that those hydrogens are then used to go through ATP synthase to make ATP. And at the very end, the electron is going to NADP plus to make NADPH. And that NADPH and ATP will be used in the next step, the Calvin cycle. And I'll make another video about that one. Bye.